This is Mark Gluck. Today we're going to talk about memory for events and for facts, what psychologists call episodic and semantic memory. Much of what we know about this field comes from the contributions of one very important man who was known for many years by only his initials, H.M. H.M. was amnesic, and his amnesia resulted from a surgery he had in 1953 when he was a young man. He had been suffering from epileptic seizures which made his life all but unbearable and unlivable, and so the doctors, to stop the surgery, took out the part of his brain where these seizures were beginning, namely the hippocampus and the areas in the middle of the brain by the temporal lobe. This cured his epilepsy, but it left him profoundly amnestic, but amnestic in a very specific way. He could not form new lasting memories. After the surgery, if he met a new doctor, if he did hours of testing with the doctor, the next day he wouldn't recognize the doctor at all. He had a favorite uncle, and when that uncle died and he was told about his uncle's death, he was very sad and upset, and he grieved. But the next day he didn't remember that it had happened, and he would ask, where was his uncle? Unfortunately, this meant that he had to constantly grieve anew each time that he learned his uncle had died. But most of his memories from before the surgery are fine. His parents' name, his childhood home, much of everything that had happened in his life up until his surgery he still retained. So it seemed as if he couldn't learn anything new in a lasting way, but he could learn some new things, but briefly, as long as he was attending to the new information. If he was given a new phone number, he could remember it as long as he repeated it over and over, but at the first interruption, the number was gone forever. He could watch a TV show and follow the plot, understand what was happening, but at the first commercial, he completely forgot the earlier part of the show and lost track. So, why was he so famous? He was so famous because we learned two very important things from studying him. One is that distinct parts of our brains play different roles in memory. Some parts of the brain are critical for encoding and learning, and others for storage and retrieval. Second, learning skills, which HM could do, uses different parts of the brain that are required for creating new memories about facts and events, which HM could not do. We'll explain more about this as we move on to the rest of the lecture. The lecture today is going to have three parts. First, we'll discuss behavioral processes underlying memory for events and for facts. The second, the brain substrates on which these behavioral processes depend. And finally, we'll look at some clinical perspectives. What happens when these brain substrates are disrupted, causing damage to the behavioral processes described initially. Let's start by focusing on behavioral processes. In particular, we'll talk about features of episodic memory and semantic memory. We'll begin with some definitions. Episodic memory is memory for personal experience of specific autobiographical events. It includes information about the spatial, where, and the temporal, when, context in which the event occurred. For example, your high school graduation day is a very salient episodic memory. You may remember what band played at the music, who you sat next to, who came and attended, the party afterwards. These are all specific events that occurred at a particular place at your high school and at a particular time when you graduated, and they're unique to you. This is different than semantic memory, which is memory for facts or general knowledge about the world. The name of the first president of the United States is an example of semantic memory. It's a fact. You may or may not remember where you first learned this. You probably don't, but it's knowledge that you and other people have about the world. So what distinguishes episodic memory from semantic memory? Well, let's look at some of the differences between them. Episodic memory and semantic memory have three contrasting properties. Episodic memory concerns specific events that occurred at a particular place and time, like your high school graduation. Semantic memories involve factual information, like the President of the United States or the first president. Episodic memory is always autobiographical. It's based on your own personal experiences. Semantic memory can be autobiographical. You can have a fact about yourself, a fact about where you went to high school. That's a semantic memory, but it need not be autobiographical. And episodic memory is always acquired in a single exposure, the event itself. Semantic memory can be acquired in a single learning event. You can be told who the first president was and remember it from that point onward, but it doesn't need to be. Lots of our memories, lots of our semantic memories come from many different repetitions. So, in general, semantic memory is strengthened by repetition. The more often we hear a fact, the more likely we are to remember it. But episodic memory can be weakened by exposure to similar events. If you've been going to lots of different graduations, your high school graduation, your junior high school graduation, your college graduation, maybe after a certain point, some of the memories of the different graduations will get confused and you'll forget who came to which. So let's see if we can test your knowledge now to see whether you really understand the difference between semantic or episodic memory. So here's the first example. A college senior takes his Latin vocabulary exam. The first phrase to be translated, is carpe diem. This is an easy one. He knows the answer is seize the day, even though he cannot remember exactly where he first heard this expression. Is the student using semantic or episodic memory? The student remembers the meaning of carpe diem using semantic memory because he has no episodic memory of acquiring this information. Let's test your knowledge again. The second phrase to be translated is netentes aut perfis. This is harder. The student can remember studying the phrase. He even recalls that the phrase was printed in black ink on the lower left of the page in his textbook, but he cannot recall the translation. Is the student using semantic or episodic memory? Here, the student does have an episodic memory of when he was first reading the phrase in the textbook. It's tagged in space and time for when he was studying this, but he has no semantic memory for what the phrase means in English. For your information, it means roughly either succeed or don't bother trying. Okay, here's a third example to test. Later in the day, the senior is helping a new student learn her way around campus. When the tour finishes, the newcomer asks where she can buy a cup of coffee. The senior thinks for a moment, then says that the coffee is better at a nearby Starbucks than at the student center. How might the senior be using both semantic and episodic memory here? Knowing that the student center serves lousy coffee is an example of general knowledge about the world, so it counts as semantic memory. Knowing how to get to Starbucks from the present location counts as semantic memory too. But why does the college senior think that the coffee is better at Starbucks? If he can remember a specific episode in which he went to the student center and had terrible coffee, or visited Starbucks and had a great experience, that would qualify as episodic memory. And so this illustrates the ways in which semantic memory and episodic memory can come together to help us function in the world. Let's talk now about encoding new memories. There are three memory processes that are involved in encoding. We begin with encoding. Then there's a storage and consolidation where the new information is made more permanent in the brain. And then retrieval, where we bring the memory out of storage and are able to apply it. So, for example, if you've witnessed this thief stealing money, and later on you're brought to the police office to identify him, and you're shown the lineup of potential suspects, and you say, no, that's not him, that's the retrieval. So you encoded the memory when you saw the thief, you stored it, you consolidated it for long term, and then you retrieved it at the point of your needing to sort of use that memory. So to repeat, encoding is the initial storage into memory, storage and consolidation is maintaining that stored memory, and retrieval is reactivating the memory for further processing. For encoding new memories, there are three basic principles that govern how successfully a new memory is encoded or stored in memory. These are 1. Mere exposure to information doesn't guarantee memory. 2. Memory is better for information that relates to prior knowledge. And 3. Deeper processing at encoding improves recall later. Let's look more closely at each of these three. Encoding, mere exposure is not enough. Unfortunately, just repetition is not an effective way of encoding new memories. For example, you've seen a penny thousands of times, but all that exposure has probably not helped you encode the visual details of a penny. Here are a whole bunch of possible pennies. Do you recognize which is the correct one? You might not. Here is what the penny really looks like. Now see if you can figure out which of these is the penny. Background context enhances encoding. Let me ask you to memorize the following sentence. If the balloons popped, the sound wouldn't be able to carry, since everything would be too far away from the correct floor. This sounds like a somewhat meaningless sentence. It's hard to understand what's going on or what they're talking about, and as a result, you'd probably have a hard time recalling it. But if before I asked you to memorize it, I showed you this picture of a man serenading a woman on the top floor of an apartment building, and he's using the balloons so that the speaker goes up to her. Now, if you were to be asked to memorize this, you understand what's happening, because we're saying if the balloons popped, the sound wouldn't be able to carry since everything would be too far away from the correct floor. Suddenly, what seemed initially like a random and confusing sentence makes complete sense when we have a context. When this was done in a psychological experiment, what they found was that presenting the picture before asking people to memorize the sentence enhanced their recall of the facts of the sentence, but the picture after the story didn't happen. So what do we learn from that? We have a study tip. Read before class, because reading after is not as effective. If you read before class, then you have a broader context within which this lecture and other lectures will make more sense, and you'll be able to understand and remember them. We'll talk now about retrieving existing memories. Encoding specificity says that more similar conditions at encoding and retrieval will affect how easily we are to retrieve something. So the encoding states that retrieval is more likely to be successful if the conditions at recall are similar to those that occurred at encoding. Here's a comic that explains this. One man says to the other, "I wonder if you'd mind giving me directions. I've never been sober in this part of town before," suggesting that his memory of being when he's drunk is specific to the different places he's visited, and when he's sober, he no longer remembers things. Now, that may seem sort of crazy, but in fact, there's some truth to that, and studies have shown that, in fact, our different psychological states, even our different physical contexts, affect where we are and how well we're able to recall. So one tip from this might be, if you know the room you're going to be tested in for an exam, you might want to do your studying in that room, because then the room becomes a state or context within which you can help recall. Another factor of recall is that more cues are better. There are many formats that are available for testing memory, formats that we use in psychological experiments or that occur in the natural world. These are free recall, a memory test that involves simply generating requested information from memory; cued recall, a memory test that involves some kind of prompt or cue to aid recall, like a little piece of a hint; and finally, recognition, a memory test that involves picking out or recognizing a studied item from a set of options. Let's experiment on yourself. Can you recall as many of the 7 dwarves as you can? Don't consult with your classmates, just use your own memory. How many can you remember? Do visual cues help? Here are pictures of the 7 dwarves. Now, if you try, maybe that'll help you remember 1 or 2. Do verbal hints help? Here's the first letter of each of their names. Does that help bring back some others? The easiest way to remember them is to be asked to recognize them, to be given a list like this that has the names of all of the dwarves from the Disney, but also other ones we call distractors. And then you go through it and ask, which of these are or are not a dwarf's name? Well, here you can see that it's a lot easier once you see them to be prompted. So there are many different ways in which we can be asked, do you remember the names of the 7 dwarfs? And these different approaches are graded in terms of how easy or hard they are. So the hardest is free recall, to generate the response from memory with no cues. A cued recall, such as the first letter or an image, that will help you a little bit more because it gives you at least one clue, one route into memory. But the easiest is recognition, where we actually see the items and we have to say, is this the dwarf or is this not a dwarf? One of the most interesting aspects of memory is when it fails, and there are many different ways in which memory can fail. We go back to our example of seeing a thief stealing money and trying to recognize who he is or who he isn't. There are many failure points. We've seen this process that goes from encoding to consolidation to retrieval. At any point along the way, memory systems can fail. There are 4 common problems with memory, 4 ways in which memory fail. They are forgetting, interference, misattribution, and false memory. And I'll tell you a little bit more about some of them. So interference is a reduction in the strength of memory due to overlap with the content of other memories. And this provides a way of distinguishing between 2 types of interference. Proactive interference is disruption of new learning by previously stored information. Retroactive interference that goes backwards in time, is a disruption of old, that is previously stored information, by more recent learning. So these proactive interference and retroactive interference are studied in the laboratory with experiments like this. So people are asked to learn two lists of paired associates. And a paired associate is where I give you one word like dog and you say chair. I give you axe and you say snow, shirt and you say tree. And then I test you on list 1. But at a later point in time, I may give you list 2, and some of the antecedents are common. So you see here that dog exists both in list 1 associated with chair and list 2 associated with window. So proactive interference is when I have trouble learning the dog window response because I still keep thinking that dog chair is the correct answer. Okay, so the interference goes from list 1 to list 2. But now if I ask you to go back to list 1 and I say, what is dog associated with in list 1? And if you accidentally keep thinking window because you're confused with what you learned more recently, that's retroactive interference. The more recent information is retroactively interfering with your first list. So you think you understand that? Let's try some examples and see if you can figure them out. Test your knowledge here on retroactive or proactive interference. I'll give you a hint. There's one of each. So the first example is Scott took 2 years of Latin in college, then spent a semester abroad in Spain, learned some Spanish while he was there, and then the next year he enrolled in another Latin course, but on his first test he mistakenly used some Spanish words instead. Is that retroactive or proactive interference? How about example 2? Marina recently broke up with her longtime boyfriend Carl. Last month she started dating Lance. One night, while she was tired and distracted, she called him Carl by mistake. Was that retroactive or proactive? So here are the answers. The first is an example of retroactive interference. Scott's new learning, Spanish, reached backward in time to disrupt his old learning. The second example is proactive interference. The old name of the old boyfriend is reaching forward in time to disrupt the new name, Lance. Another type of memory failure are misattribution errors. So a common memory failure is we incorrectly link memories to their source. And there are several different types of errors that are possible from this misattribution. One is when the information is correctly remembered, but mistakenly associated with an incorrect source. Joe told me the party was here, that's what I think, when it was actually Jane, so I confuse who I attribute this information to. Source amnesia is when the information is correctly remembered, but the source is not remembered at all. For example, I know the party is here, but I have no idea how I know that. Cryptomnesia is mistakenly remembering someone else's ideas as one's own. I think it was my idea to have the party, when actually it was Jane's idea. Cryptomnesia is a very interesting phenomenon, and it crops up in a number of circumstances, many of them having to do with intellectual property or intellectual priority. To understand that, let's listen to 2 songs from years ago. The first is the Chiffon song He's So Fine from 1962.
Do-lang, do-lang, do-lang. Do-lang, do-lang. He's so fine. Do-lang, do-lang, do-lang. Wish she were mine. Do-lang, do-lang, do-lang. That handsome boy over there. The one with the wavy hair. I don't know how I'm gonna do it, but I'm gonna make it mine. Be the envy of all the girls. It's just a matter of time.
So that was 1962. Let's go ahead 8 years to 1970, when George Harrison came out with a song that ultimately became much more famous, My Sweet Lord.
My sweet Lord, my Lord, my sweet Lord, I really want to see you, really want to be with you, really want to see you, Lord, but it takes so long, my Lord.
So Cryptomnesia. Did those 2 songs sound familiar to you? Did they sound similar to you? Do you think that one was a copy of the other, the later one? Well, the producers, the publishers of the first song did. On February in 1971, Bright Tunes Music Corporation filed suit alleging that the George Harrison hit My Sweet Lord was a plagiarism of He's So Fine. If you were the judge, what would you rule? Do you think Harrison plagiarized or not? Well, the judge ruled in the suit in favor of Bright Tunes, determining that Harrison had committed subconscious plagiarism and awarded them over $500,000 in damages. So by subconscious plagiarism, what they meant was that Harrison didn't necessarily consciously try to plagiarize it, but he may have sort of been hearing the sound, the song in his memory, and thought mistakenly that he had developed this song himself. At least that's what the judge described or believed. There's another type of memory failure called false memory. It's related to the phenomenon of source memory, and it's memory for events that never actually happened. In laboratory research, false memories are particularly likely to occur when people are prompted to imagine missing details, and later they may mistakenly remember those details as the truth. This is often what happens in eyewitness testimony, where a detective or a prosecutor is pressuring someone to say, do you really think this is the person? Are you sure it's not the person? Are you sure you can't remember it? And under these circumstances, people often come up with details and begin to believe something that never actually occurred. One study reviewed 62 cases in which people were convicted of crimes and later exonerated based on DNA evidence that showed they absolutely could not have committed the crime. In more than 80% of these cases, the crucial evidence leading to conviction was eyewitness testimony, in which witnesses had mistakenly identified people later who were proven to be innocent. We can see some examples of creating false memories, not only in the laboratory, but in a study that was done by Slate magazine. They tested people, their readers, who were fairly sophisticated about the world, about whether or not they remembered certain events. And most of these events that they showed them were real, but some were doctored. You see here on the left is a photo of former President Obama with some unknown person, and they doctored the photo so it looked like he was with the president of Iran. And when they mixed this in with lots of other real photos from history and asked people if they remembered the event, sure enough, half of Slate's magazine readers misremembered seeing the doctored photo on the right of Obama with the president of Iran. It was a false memory. So let's summarize what we've learned so far about the behavioral processes of memory for facts and events. First, episodic memory is our memory for specific events that we experienced in a unique spatial and temporal context. It is information we remember. Semantic memory is memory for facts about the world and personal information about ourselves. It does not necessarily include information about where or when the memory was originally acquired. This is information we know. So you can think about episodic and semantic as the difference between what we remember versus what we know. Both episodic and semantic memories can be flexibly communicated in ways other than those in which they were originally acquired, and both are available to conscious recollection. Once we've learned something in one way, we can describe it in another way. It's memories that we can actively use and apply and understand and think about. Several principles govern the encoding of new memories. Mere exposure to information does not guarantee memory, like looking at pennies. Memory for new information is stronger if it can be related to existing knowledge, like the context in which we saw the balloons floating with the speaker. And memory is also affected by how deeply the information is processed during encoding. The more we think about information, the more we work on it, the more likely we are to remember what's happened. Successful retrieval of existing memory is more likely if, one, encoding and retrieval conditions match, this is called transfer-appropriate processing, and two, if more cues are available to prompt recall. Memory can also fail in many ways due to simple forgetting, interference, and creation of false memories. Source amnesia is a related phenomena in which we remember information but are mistaken about where or how we learned it. We'll now talk about the next part of this lecture, which is brain substrates. What are the brain mechanisms that underlie the behavioral processes which we were just discussing? And we'll begin by talking about cortical networks for semantic memory. There are two types of cortical networks that are important for memory. The sensory cortexes, the cortices, these are areas of the cerebral cortex involved in processing sensory information, such as sight and sound. And association cortex, which are areas of the cerebral cortex involved in associating information within and across the sensory modalities, essentially where information from different senses are integrated. Semantic memories seem to be stored in a distributed fashion throughout the cerebral cortex, so that the first cortical processing centers for a sense would be in the different sensory modalities, the visual cortex or the auditory cortex. And then the association cortex would link across these various areas. Taken together, this evidence suggests that semantic memories are stored not in a single place in the brain. There is no particular part of the brain that has a memory for your grandmother, but rather memories, such as your grandmother, are stored across the many specialized processing centers in the cortex. How she looks would be stored in the visual cortex, your memory of her perfume might be stored in the olfactory cortex, and the sound of her voice in the auditory cortex. Again, an example, your semantic memory of an apple would involve visual components in visual cortex, olfactory components in olfactory cortex, and so on and so forth. So the idea is that the memory doesn't exist in one place, but is distributed throughout the brain. Semantic memories seem to be stored in a distributed fashion throughout the cerebral cortex, and Penfield, a famous researcher, found that cortical stimulation could evoke simple sensations and sometimes complex memories depending on where they were stimulated. Cortical lesions can display different types of agnosia, a selective semantic memory impairment. So auditory agnosia for speech, it's certain damage to certain parts of the brain will prevent someone from being able to understand spoken words, although other sounds are recognized and reading and writing is intact, suggesting that there are specific parts of the brain that are involved in understanding spoken words. Tactile agnosia are brain damage that can result in an inability to recognize an object by feel. You could see an object and know what it is. It's a hammer or it's a cup. But if your eyes were closed and you were given it to feel, you wouldn't be able to recognize it. Other studies from recordings in human cortex show that neurons can be tuned to specific semantic categories. Memory representation in cortex. This was a study that was done that had repeated presentations of one face, A, and another face, B. And what they found was that patterns of A, whenever they appeared, the pattern of activity done in the left fusiform gyrus was part of the visual association cortex, that the patterns, whenever the A faces were presented, looked a lot more like each other than the patterns in the B faces. And the more similar the different presentations of A the more similar the neural representations in this brain region when A's were presented, the more likely people were to recall it, suggesting that a coherent and constant representation in the brain of a particular pattern was associated with being able to remember that and later recall it better. A particular area of interest, one that we heard about earlier when we talked about HM, are the medial temporal lobes and memory storage. So the medial temporal lobes are in the middle of the brain, behind the temples, just above your ears. And part of the medial temporal lobe, one of the most critical parts, is the hippocampus. So how do these new memories get stored and consolidated in cortex? Consolidation seems to depend on the medial temporal lobe, often referred to as MTL, which includes the hippocampus and the surrounding cortex, the entorhinal cortex, the perirhinal cortex, and the parahippocampal cortex, all of which were the brain regions that HM had removed. So here we see a diagram that shows where the hippocampus is, the blue structure, and then you see if the brain is sliced along the plane that's shown there, that you can see that there's the hippocampus in blue, and then these other surrounding cortical areas, the entorhinal cortex, which is right next to the hippocampus, is the pathway through which information enters the hippocampus and leaves the hippocampus, and other areas, the perirhinal cortex and the parahippocampal cortex, which are all part of this brain region that is processing the encoding of new memories. Now, some of the evidence for this comes from lesions of the medial temporal lobe. They produce anterograde amnesia, the inability to consolidate new memories, new declarative memories, memories for facts and events. And this is what we saw earlier when we heard about HM. Hippocampal activity also predicts subsequent ability to remember. The more hippocampal activity there is during a learning experience, the more likely we are to recall it later. Let's go back now to learn a little bit more about HM and hear from Brenda Milner, the psychologist who studied HM for many years.
It was one such test, the mirror drawing test, that revealed to Dr. Milner evidence that the human brain had more than one memory system.
For all of us, you or me today, whenever you get to the point of the star where you have to change direction, your hand does this kind of thing, but then by doing, not by telling yourself what you ought to be doing, but just by doing in some way, by practice, you gradually improve and you keep a nice smooth line, and at the end it's beautiful. That is what you and I would do. but it's also what HM did. HM, over the 3 days that I tested him, and I have a lovely learning curve for this, that he got better and better, and he was doing beautifully at the end. And he had absolutely no recollection of the 3 days training he'd done on this. It was, that one trial was unique to him, experience, and he was proud of himself that he had done it so well. No recollection at all of all that had gone before. And it was this dissociation, you know, between his total lack of, between what he was aware of and what he had achieved. That was, to me, very exciting. If anybody asked me, you know, the really exciting moment, it was actually then, you know, looking with my eyes very wide at this going on. And this was early evidence that there's more than one learning and memory system.
So what we learned from this study is that HM's skill learning, his ability to learn a skill like complex tracing, is intact, even though his episodic memory for the times when he learned that is gone and missing, and suggests that those are different pathways in the brain. The skill learning takes place unconsciously, out of awareness, but the conscious awareness of the events when you learned it is separate, and that depend on the hippocampus and the other brain regions that he lost. So there was another patient known as EP, and he also had bilateral damage to the medial temporal lobe, so he was similar to HM. And this shows some data. There's a classic task here where we show someone an original figure, and they then have to copy it while they're looking at it, and most people can do that. But other people can copy it. That's what's shown at the top for a healthy control. But then 10 minutes later, they come back and they're said, now tell us what you saw. You know, tell us what this figure looked like. And they can get most of it correct. You can see a healthy, normal person after a 10 or 15 minute delay. Doesn't get it exactly right, but gets most of the features there. EP, on the other hand, could copy just fine. If the original figure was present and he could see it, he could copy it well. But 10 minutes later, he had absolutely no idea what he had seen and no recollection of it. So he can directly copy it to an intact working memory, but he can't draw the figure from memory after a short delay. We have a unique video next, which is with the Alan Alda, who you may remember from MASH, Larry Squire, a psychologist from UC San Diego who studied EP, and a chance to actually meet EP yourself.
We're starting our exploration of memory a few miles up the coast from La Jolla, with a man whose own ability to create memories vanished almost entirely a decade ago. Ever since, he's been helping researchers from the University of California, San Diego.
Hi, good to see you, by golly.
Today, Larry Squire and Jen Frascino are on the latest of many visits the team has made to the man known to memory researchers everywhere as EP.
Oh, it's wonderful. Got a nice sun for the camera.
Yeah.
A little work?
We're going to go in, you're going to do some work with Jen.
EP is now 82.
This is fun, enjoyable.
In 1992, retired from a career in electronics, he suffered an acute virus infection that destroyed part of his brain.
Research and development, that's what I like.
Most of his thinking skills survived the damage virtually intact.
Why should people pay taxes?
To support your country and your counties and states to pay for things.
If you were lost in the forest during the day, how would you go about finding your way out?
I would find a river and follow the river down to wherever it may go. Ocean, lake.
EP can also copy complex drawings.
About it, huh?
And he not only has no difficulty repeating back a list of words, in this case, dog, raccoon, lion, and parrot.
Dog, raccoon, lion, and a parrot.
Good.
But he even has a little sly fun with Jen along the way.
What you gotta add is about 2 more.
Oh, really?
Yeah,
Would that make it tougher?
Then that would catch them. 7, you know. Leaf. Medicine.
EP really hits his stride when the testing of his cognitive skills involves Jen's laptop computer.
Gold. Having been in electronics over the years, I am amazed at all this. It's just wonderful. Believe me, little printed circuits, just, oh, you can't believe it. It used to be in a 6-foot rack, and down to this is just wonderful. When I was in electronics, this would be in a 6-foot rack.
But it's now that something odd about EP's behavior becomes apparent.
Printed circuits, diodes, triodes, and all that stuff in there. Oh, it's just amazing, believe me. When I was in electronics, this would be in a 6-foot rack. This would be in a 6-foot rack. It would be in a couple 6-foot racks. It would be in 6-foot racks. This would be in a 6-foot rack. And now look at it, down to this. Little printed circuits and...
So he's not aware that he's told us 3 or 4 times that he used to be in electronics, that a computer used to fill a room, and that now it's down to the size of a microchip. He's just not aware that he said those things several times.
No, he tells us the same stories again and again. We've heard these stories before, and we'll hear these stories again.
The equipment that it takes.
How was it? How have you been enjoying this session?
I enjoy this.
Yeah,
Absolutely.
It is interesting, isn't it?
Look at the work you guys are doing. You're getting wonderful, I believe.
I'm sorry, I forget her name. What's her name?
Her name?
Yeah.
I don't remember.
Okay, Jennifer.
Jennifer.
Yeah.
I'm sorry.
Have you met Jennifer before today?
I think so, yes. I'm not positive, though.
Yeah.
Okay.
I'm sorry, my...
No.
Oh, that's
It's okay.
Yeah.
We all have.
Well, we all get that one.
Oh, sure. How much have you worked with him?
We have probably visited his house 200 times.
200 times. And for him, is each time the first time for him?
Well, you see, it's yes and no. It's clear that he does have a record of the experiences that are based on the habits, the interactions, the emotional values of the interactions, the fact that they've been positive and not negative. He certainly lets us in the door faster.
I mean, like you saw, he couldn't tell you my name.
Yeah.
He couldn't tell you if you saw me before. But there's definitely a rapport there that I think has been built, and he's aware on some level. I read you some words about 15 minutes ago. 4 of them. 4 words. Do you remember me reading you 4 words?
No, I can't
No?
recall. I'm sorry.
No, that's okay. Don't need to
My
apologize.
mind is getting old.
That's okay. I read you 4 animals, and I want you to tell me yes if you think the animal was on the list I read you about 15 minutes ago, or no if it was not on the list.
Wonderful.
Okay? Cat.
Yes.
How sure are you?
I feel reasonably sure
Reasonably
on that cat.
sure? Uh-huh. Raccoon.
No. I feel no.
Giraffe.
Yes.
How sure are you that I read
I'm
you that
real
word?
sure on that.
Real sure?
They're beautiful animals,
Okay.
believe me.
Lion.
No.
Okay.
EP's inability to remember things that happened just a few minutes ago can be traced directly to the damage the virus inflicted on his brain a decade ago. The areas destroyed were in his temporal lobes, most critically a structure called the hippocampus and the brain tissue immediately around it. Without a hippocampus, EP lives in a permanent present. Anything new that happens to him simply doesn't get recorded.
Do you remember drawing something for me today?
No.
Drawing something about
No.
15, 20 minutes ago?
No.
Remember picking? Okay.
I'm sorry.
I don't apologize.
Yeah.
Thank you for trying. Thank you.
But although EP can't record new memories, old ones from the years before his hippocampus was destroyed are remarkably intact.
Hayward.
He grew up in Hayward, California.
How about your house on Castro Valley Boulevard to the Hayward Theater? Remember where the Hayward Theater is?
Yes, I would leave the house and turn to the right to A Street, down A Street to Castro Boulevard, and turn to the left, and it's in the middle of the block. Hayward,
Okay, and if you didn't go down to A Street, if you instead you could take another street. What would
the
you do?
Center, Redwood Road.
Okay.
Redwood Road would run right into it.
Unbelievable.
How about the public library, Hayward Public Library, from your house?
Be the same thing going down A Street, and then A, you have to go to the left to B, and it's on the corner. I don't
He's got that map in his head.
I know how
He
to
can
get
see
there,
all
but
these
it's
streets.
the boulevard.
He used to ride his bicycle around.
The last thing I want to talk to you about is the neighborhood that we're in now.
Here.
How about any of the streets around here? Can you tell me any of the street names in the neighborhood here?
No, I cannot.
That's hard. That's a tough one.
Yeah. I'm sorry.
No, you don't need to apologize.
I know where Castro Valley Boulevard, but that's where I was
Absolutely.
born and raised. But right
Absolutely.
here, I have to say no,
No.
I'm sorry. No, I just live right here.
Enjoy the world
So I
rather than
enjoy our
driving.
area right here.
Okay.
You seem to enjoy your life.
Oh, I have.
The enthusiastic help of amnesics like EP has pinpointed the hippocampus as crucial for recording new memories.
Those places, it was just wonderful.
At the same time, the fact that EP has vivid memories from years before his hippocampus was destroyed means the hippocampus can't be where they're permanently stored.
We love it here.
So now you've seen and heard about two people who lost their memory, lost their hippocampus, and from their hippocampus lost the ability to form new memories. HM, who has since died, and EP, who is still alive. But what if the other thing had happened? What if you had a big hippocampus? Would that help you? Well, one study was done in London on taxicab drivers, and taxicab drivers in London have to do an incredibly detailed study called The Knowledge, where they spend many years learning every possible route around London. This, of course, predates Google Maps and smartphones, but they had to know in their head how to make their way all around London, and they would be tested, and they could only get their taxi license once they got it. So the question is, do these taxi drivers who have this incredible spatial knowledge of London, do they have a bigger hippocampus than other people of the same age? And in a study done, it showed that, in fact, they do, that licensed London taxi drivers have a slightly larger hippocampus than those who are normal controls. Now, this doesn't tell us what's cause and effect. Is it the case that having spent many years studying the layout of the streets of London and being a taxi driver, does that grow your hippocampus? Or is it just the case that people who have a bigger hippocampus are more likely to pass the exam and become London taxi drivers? So this is a correlation. It doesn't tell us which is the cause and which is the effect. Another insight into understanding the role of the hippocampus in memory comes from another study of paired associate recall, which looked at the similarity of activation patterns in the medial temporal lobe during encoding and retrieval. And so, again, just like before where we saw dog and window and the other paired associate, this is a paired associate training where the associates are images. So you'll see at the beginning there is a rubber ducky and a rose, and you have to learn that when you see the rubber ducky, you have to respond with rose. And later on, you're tested, you're shown the rubber ducky, and you have to give the response that it's a rose. Now, what's interesting is we can look at what happens in the brain when you're shown the rubber ducky during the encoding, when you're learning, and later on when you see the retrieval. And what the investigators found is that the similarity between the patterns of activity in the hippocampus, in the medial temporal lobe, in fact, more broadly, is greater when there is successful retrieval. So the idea is that the pattern of activity during retrieval, if that's activating the same pattern for rubber ducky as when you were encoding it and learning it the first time, you're more likely to know whether that's associated with the rose. And we see that both in the hippocampus itself as well as in the perirhinal cortex, which is the area that's just around the hippocampus that we saw in an earlier diagram. So let's talk a little bit more about memory and the medial temporal lobes. Another study that gets at this question of what's the role of the medial temporal lobes in memory is called a subsequent forgetting paradigm. So participants are scanned while learning a list of words. They're later tested on words, and we can go back and compare the brain activity for words that were remembered versus words that were forgotten. So the idea is that we're scanning their brain while they're learning the words, and then later on, some of those words are remembered, some of those words are not, and we go back and we ask, what's the difference in the brain between the words that when they first saw them, that they later learned, that they later remembered, and those that they later did not? And what it showed is that indeed there are two brain regions, the medial temporal lobe, which is what we've been talking about, and also the frontal lobe, a brain region which is involved in executive control. So good encoding and consolidation for long-term memory activates the hippocampal area of the temporal lobe and also the left frontal cortex. So what determines what does or doesn't get stored? Let's look at that. We saw before some data suggesting that activation of the left frontal cortex is involved in accurately encoding information that's later correctly recalled. The frontal cortex are those regions of the cortex that lie within the frontal lobes, and they help determine what information we store and what information we forget. And you can see here where the frontal cortex is as compared to other parts of the brain, including the hippocampus. Experiments strongly suggest that the frontal lobes help determine what new information gets encoded as episodic and semantic memories and what gets forgotten. How does it do this? Well, we don't know exactly, but we think the frontal lobes may be critical for binding the contextual information to the event memory. So the ways in which all the various contexts, where you were, when you were, what you were smelling, what you were seeing, that the frontal cortex is critically involved in tying all of these together. Another brain region involved is called the basal forebrain. The basal forebrain is a collection of structures that lie at the base of the forebrain and are important in the production of acetylcholine, a chemical that's then distributed throughout the brain. So another way that you can have a memory loss is not just by damage to the hippocampus or the frontal lobes, but by damage to the basal forebrain. One feature of basal forebrain amnesia is that when questioned about past events, patients may respond with highly detailed but false memories, a phenomenon called confabulation. Confabulation typically appears in patients in whom the damage from an aneurysm rupture extends beyond the basal forebrain and into the frontal cortex. So this suggests that anterograde amnesia, the inability to form new memories, can result from many different types of brain damage, each of which are disrupting a circuit, a whole set of circuits that are involved in forming new memories. The last part of the brain substrate section is understanding something about long-term storage and retrieval. The general view of how we remember and store information is called the standard consolidation theory, and it holds that the hippocampus and related medial temporal lobe structures are required for the initial storage and retrieval in episodic memory, but that their contribution diminishes over time until the cortex is capable of retrieving the memory without hippocampal help. This view conceptualizes episodic memories consisting of many components. We see here that initially memories are distributed. We saw this before, this idea that the different sensory cortex, there's no grandmother neuron for your grandmother, but there's the vision of her and the smell of her and the look of her and how she sounds in different parts of the brain. And that initially the hippocampus is involved in your storing all those different parts of your memory for your grandmother. But after a time, the hippocampus is no longer critically involved, and your memory for your grandmother exists independent of your hippocampus. And this is very much why we saw with HM and later in the video with EP that once these memories have become consolidated and are dependent only on the cortex, then EP and HM don't need their hippocampus anymore. And that's how EP was able to have incredibly exquisite recall for the neighborhood he grew up with as a child. Retrograde amnesia is a different sort of amnesia. Retrograde amnesia is amnesia for what happened before an accident or before a particular time. And in the late 1800s, Theodore Ribot noticed that individuals with head injury often develop retrograde amnesia, that is, the loss of memories for events that occurred before the injury. And typically, retrograde amnesia follows a pattern called the Ribot gradient, in which retrograde memory loss is worse for events that occurred slightly before the injury than for events that occurred in the past. So patients with bilateral medial temporal lobe damage generally show some retrograde amnesia along with their anterograde amnesia. And we'll look at a schematic of this next. So what we see here is that there's the injury between the sort of orange and purple parts, and a healthy person and today is shown on the far right, so the injury occurred somewhat in the past. And what we see for a healthy person, shown in blue, the further back you go in time, the less you remember accurately, and that's normal. An amnesic patient, like HM or EP, shows profound inability to remember anything that happened between the injury and today. So where EP lived or who they met, that's all gone. But there's also a little bit of retrograde amnesia, which is that the injury also causes some impairments to things in the past, but only really the things that happened right immediately before the injury. And why do you see that Ribot gradient, that red line going, slowly back and seeing some impairments before the injury? That's probably because the hippocampus is still involved in consolidating the memories. So the idea is that during that period where we see this retrograde amnesia, That's the period prior to the injury where the hippocampus was still important for tying together the various parts of memory in the cortex. But once that period has gone beyond then, once memories are further older than that, then the memories are still quite intact. So let's test your knowledge about retrograde versus anterograde amnesia. Now remember, before we talked about interference. Now we're going to talk about retrograde and anterograde amnesia. So I'll ask you a couple of scenarios, and you should see whether you think this is retrograde or anterograde amnesia. 10 years ago, John was involved in a motorcycle accident that caused damage to his medial temporal lobes, including his hippocampus. He spent 2 weeks in the hospital, recovered before being released to the care of his wife at home. Now John has no memory of the time he spent in the hospital. Is that retrograde amnesia or anterograde amnesia? This is anterograde amnesia because even though the event, the hospitalization, happened a long time ago, it happened after John's brain injury, and the hippocampal damage at that time prevented him from forming a new memory of it. John also has no memory of events that occurred in the months before his accident, although he remembers his childhood in Minnesota. Is this retrograde or anterograde amnesia? This is retrograde amnesia. John's injury reaches back in time to disrupt memory for events that occurred before his injury, but not so long before that they affect things way back in his childhood. So this is the Rebo gradient, the idea that events that had occurred just prior to the injury or the accident are still not stable without the hippocampus. And if you remove the hippocampus, you disrupt those recent memories. Another important aspect of memory is sleep. Sleep is critical to memory formation, and a large body of research suggests that sleep plays an important role in how memories are encoded in the hippocampus and then eventually get stored in cortex. Over the course of the night, the brain goes through several stages of sleep. Rapid eye movement, or REM sleep, is a phase of sleep in which the eyes move rapidly under closed lids. Non-REM sleep is a category of sleep that includes both light sleep and slow-wave sleep. And slow-wave sleep is a phase of sleep characterized by large, slow oscillations in the brain that are highly synchronized over wide areas. And it is this slow-wave sleep that's believed to be particularly important for the encoding of memories, for taking the memories that are in the hippocampus and transporting them and mapping them into areas of the cortex. So slow-wave sleep is deemed to be a critical component of this consolidation process where memories in the hippocampus are put out to become more permanent in cortex. In spontaneous memory reactivation is the idea that in the hippocampus during slow-wave sleep, networks of neurons that were active during the day become active again, seeming to rapidly and spontaneously replay activity patterns as if they are experienced during the day. Research suggests that such rapid replay during sleep may be an important mechanism for memory consolidation. These studies, although some of them have been done in humans, most of them have been done in animals where we can actually record from the various cells. And what you see is that as the rat is going across through an environment shown here by these rainbow colors, different cells are firing as the rat moves from one place to another, these place cells in the hippocampus. And what happens when that rat is sleeping is that same pattern of activity from the blue to the green to the yellow to the red goes in that same sequence, but it happens very rapidly. So it's as if what's happening during this slow-wave sleep is a rapid replay of memory, and it's believed that that rapid replay is critically involved in taking the memories that are in the hippocampus and during sleep rapidly transitioning them to the cortex. So let's stop for a moment and summarize what we've learned about the brain substrates of event and fact memory. Semantic memories seem to be stored in the cortex. To some extent, specific kinds of semantic information are stored in the cortical areas devoted to processing that kind of information. So visual memories in the visual cortex, auditory memories in the auditory cortex. The hippocampal region, which lies in the medial temporal lobe in humans and other primates, is important for new episodic memory formation. Patients with bilateral hippocampal region damage show anterograde amnesia, that is, the loss of ability to encode new episodic and semantic memories, and they may also show retrograde amnesia, disruption of old memories acquired before the damage. In healthy humans, functional neuroimaging shows that the hippocampal region is especially active during encoding of information that will be successfully remembered later. Standard consolidation theory states that older memories are stored in cortex and eventually become independent of the hippocampus. The frontal cortex is important for determining which new memories are stored or forgotten, as well as for remembering the source of information. Other brain areas involved in episodic and semantic memory include the basal forebrain, which may help guide how the hippocampus stores new information. In the final part of this lecture, we'll talk about clinical perspectives. What are some of the ways in which these brain substrates for events and for fact memory can be disrupted, and how does that affect the behavioral processes that we can see as symptoms in these patients? We'll start by talking about transient global amnesia. Transient global amnesia is transient, that means it's temporary, disruption of memory not due to known causes such as head injury or epilepsy. Transient global amnesia is difficult to study precisely because it does not last long. Studies using neuroimaging methods suggest that many individuals experience an episode of TGA show small abnormalities in the hippocampus. In some cases, these abnormalities are no longer detectable on follow-up imaging after the TGA has resolved. So here's some data here. In the hours since a TGA, both anterograde and retrograde amnesia subside. So it suggests that whatever is happening in transient global amnesia, it has both some anterograde and retrograde components, but that these resolve after a period of time, suggesting that perhaps there's a transient disruption in the hippocampus. Functional amnesia, sometimes called psychogenic amnesia, is a sudden, massive retrograde memory loss that seems to result from psychological causes rather than physical causes. Imaging results suggest that functional amnesia may reflect dysfunction of the same brain regions that are damaged in organic amnesia. So to summarize, organic amnesia is brought on by a physiological injury, such as the surgery or the virus that HM and EP had, while functional amnesia is brought on by psychological trauma. So these are two very different types of amnesia. And this gives us an opportunity to look at various ways in which amnesia has been presented in the popular media, in movie and TV. And so we're going to have a bit of a test yourself here, where I want to ask you two questions about each of the movie clips that I'm going to play. For each of the following movie or TV clips, indicate if the character has anterograde or retrograde amnesia, and if you think from the bit of the plot that you've been able to see, whether this origin is organic or psychogenic.
Number 3, Dr. Anthony Edwards or John Ballantyne, Spellbound.
I have no memory. It's like looking into a mirror and seeing nothing but the mirror.
An Alfred Hitchcock classic, this psychological thriller stars Ingrid Bergman and Gregory Peck as a pair of physicians looking to solve a murder/identity mystery.
And if we can unlock one tiny memory, it'll give us a key to the others.
Peck plays a man who can't remember his true identity and has assumed that of a doctor named Anthony Edwards, who believes that the real man with the same name has been killed.
Remember now. Edwards is dead.
Bergman is a psychoanalyst who doesn't believe he was responsible for Edwards' murder. And the two utilize methods of psychotherapy to uncover.
So, do you think that was retrograde or anterograde amnesia? And was the origin psychogenic or organic? The answer is retrograde amnesia of a psychogenic origin. Retrograde because he forgets who he was in the past, and psychogenic because it comes from a trauma, a psychological trauma for which they're trying to use therapy to uncover. Let's move to the next one.
I know who I am. I know all about myself. Since my injury, I can't make new memories. Everything fades. If we talk for too long, I'll forget how we started. And next time I see you, I'm not going to remember this conversation. I don't even know if I've met you before.
So, does that line sound familiar? That's not so different than what we saw EP saying to Alan Alda and Larry Squire. So anterograde or retrograde, organic or psychogenic? Well, like EP and HM, the character in the movie Memento had anterograde amnesia. He can't remember new things, and he referenced an accident that he had, and so it was of an organic nature, not a psychological trauma. Next movie is The Bourne Identity.
I can't remember
2002.
anything that happened before 2 weeks ago.
Amnesia?
Yes.
Is to find out who he is.
I guess you're not home. Monsieur Bond!
I don't recognize any of this. I don't recognize any of this.
Before they find him.
So, and the answer here is retrograde amnesia of an organic nature. Namely, he had an accident earlier on, although that may not have been evident here. So, we have one last one. It's a TV clip from many, many decades ago. I Dream of Jeannie.
And I'll never forget what's her name. I Dream of Jeannie.
I remember.
In TV land, one minor blow to the head automatically results in amnesia.
Did you hurt yourself?
After a vase hits Tony in I Dream of Jeannie, he immediately forgets about the mystical lady he unleashed from a bottle.
Major Nelson hit his head and now he does not remember me.
You mean he has amnesia? Huh.
Okay, retrograde, anterograde, organic, or psychogenic? Well, this is retrograde amnesia. He forgets what happened in the past. He forgets who Jeannie is. And it's organic because he was hit on the head. So how well did you do on those 4 movie clips? If you got all or most of them right, then you've probably mastered the concepts of amnesia. So let's go now and talk about the summary of what we've seen from these clinical perspectives. First, transient global amnesia is a temporary disruption of memory that occurs without evidence of head injury or epilepsy. However, there may be precipitating events such as vigorous exercise or stress. During a TGA episode, patients typically show severe anterograde amnesia and usually some degree of retrograde amnesia. TGA may reflect a temporary disruption of brain function due to interruption of blood and nutrients to the brain. Typically, as the brain recovers, memory returns to normal. Functional amnesia is a very rare form of amnesia, except in TV and movies, in which patients lose memory for specific episodes in their lives or even forget their entire identity. Functional amnesia seems to result from psychological trauma rather than any physical or organic cause. So this brings us to the end of the 3 main sections of this lecture. What are you responsible for? What do I hope that you learned? What are you likely to be tested on in the exams to come? Let's review the learning objectives for each of the 3 sections. For the behavioral processes, you should be able to compare and contrast episodic and semantic memories, describe how encoding is influenced by mere exposure, explain why memory is better for information that relates to prior knowledge, describe how depth of processing enhances memory, describe the concept of transfer-appropriate processing, describe how interference accounts for forgetting, and distinguish between retroactive and proactive interference, describe the memory failures of misattribution, source amnesia, cryptamnesia, and false memory, and describe the concept of a consolidation period. For brain substrates, you should describe the role of the cortex in memory formation and storage, be able to describe the role of the medial temporal lobes in memory storage, the role of the frontal cortex in memory storage and retrieval, and the roles of the subcortical structures in memory. For clinical perspectives, you should be able to explain how transient global amnesia incorporates both retrograde and anterograde amnesia and why it is so difficult to study. And second, you should understand the distinction between psychogenic and organic amnesia. So this brings us to the end of the 3 main sets of learning objectives that you should have acquired from listening to this lecture and reviewing the material. Now, of course, you'll need to study for the exam and for the quizzes to come. So let me give you a little bit more information about what's the neuroscience of learning and memory that can help you study
Pulling
smart.
an all-nighter to study is like taking a gallon of water and thinking if I keep pouring it into this 6-ounce cup, I'm going to have more and more water, but you've only got this much that you can hold at a moment. Learning is the process by which we acquire new memories and new skills. This is the science behind memory.
Some people can cram and just like get an A on the test. Like, I can't do that.
Every single time I wanted to study more, I kept on falling asleep, so it didn't really work out.
The way short-term memories become long-term memories has to do with a dialogue between two critical players in the memory game. One is called the hippocampus, and the other is the cerebral cortex. If we don't sleep, then all the things we have in short-term memory will stay there, and they'll never make the trip from the hippocampus to the cerebral cortex. And that's why sleep is so critical to memory.
During class, I take the notes, and then I go back into the textbook, read the material that's new, and try to repeat the problems that are given to me.
I usually study for a test up until the test, like regardless, like no matter how long I've been studying.
Tips for learning quickly are the same tips for any sort of learning. One is your body and your brain have to be ready. You have to be well slept, low stressed, and cardiovascularly fit. The best way to learn is to do two things. One is to pace yourself.
I switch subjects like on and off. I go from like one subject and then I take a break and do like another subject.
You need the breaks in between for the memories to get organized and integrated with the rest.
I need a lot of repetition to the point where like I make like flashcards, I rewrite my notes. So like I just keep like reading the notes, rereading the notes until it like sticks in my head.
You want to do not just sort of rote repetition, you want to repeat things in context. You want to elaborate on the memories. You want to go into depth. And the last tip is you want to be quizzed, you want to be questioned, you want to have novel questions similar to the kinds that you might encounter on the exam as you have during the studying.
Okay, so thank you very much. This brings us just about to the end. And I want to thank you all for joining me for this lecture. And I hope you remember it well.
We met at 8.
I was on time.
No, you were late.
Ah, yes? I remember it well. We dined with friends.
We dined alone.
A tenor sang.
A baritone.
Ah, yes. I remember it well. That dazzling April moon.
There was none that night, and the month was June.
That's right. That's right.
It warms my heart to know that you remember still the way you do.
Ah, yes. I remember it well.